It's my pleasure to introduce our final speaker of the morning, Andrew Hipp. Last night he shared a little bit of this infamous phone call where I called him about wanting to learn something called AFLP. And I had just heard that as a buzzword from another expert, and we talked, and suddenly Andrew's life changed, and so did mine. And I think in both cases, for the better. Andrew always calls me Dr. Rothrock, which just tickles the heck out of me. But we did have some, I've always appreciated his combination of field work, traditional taxonomy, and laboratory expertise, computer analysis, just an amazingly multifaceted scientist, but also just deeply thoughtful, cultured, with a lovely family. He loves his two boys to death, and total mensch. So, Andrew, I'm looking forward to what you have to share with us this morning.
Thank you, Dr. Rothrock. It's a real honor to be here to speak with you today, both because I have such high regard for Paul, and because it's such an exciting group. Both the Sedges, but here I'm actually talking about the people. It's such a wonderful group to be here with, and I feel humbled and happy. Anyway, I'm going to be talking with you today about. There we go. I was asked, oddly enough, so Paul, in giving this talk, suggested that I speak about oaks, and I was afraid that if I did that, I didn't think I would be stoned, but I was afraid that people would be unhappy, because this is a Sedge conference. So I said, well, what if we talk about both? Because I work a lot on sedges and a lot on oaks, and they have a lot to tell us about what we mean when we talk about species. As a systematist, a lot of the questions that I have revolve around what in God's name do we mean when we say things like Carex stricta? What do we mean when we say Quercus rubra? I'm not going to answer that question today, but I am going to suggest insights that you gain by studying sedges and oaks, which really lie at different ends of the plant world. Let's see if this will work. Coming in. There we go. So sedges and oaks are very different in some striking ways. Oaks are long-lived, outcrossing organisms, often studied with binoculars. These things can live to be hundreds of years, whereas individual sedge shoots only live a single year, typically, maybe two. They're small. They're often selfing. So they behave differently in many ways, but they both exhibit striking diversity, both what we can see from a distance and when we look up close. The magnificence, ecological diversity, morphological diversity of sedges and oaks begs the question of how we got so many of them, right? Why are there so many species? And they still operate from different ends of the plant world. In oaks, where we've got about 425 species worldwide, here I'm talking about the genus Quercus. I'm limiting myself to the genus Quercus, genus Carex. They're typically outcrossing, high rates of introgression. They've been termed hybridization, lots of movement of genes between species. They've been termed a worst case scenario for the biological species concept, if you want to adhere to that in the first place. So they beg the question, are species even distinct in oaks? Do we have species? And sedges, on the flip side, about 2,000 species worldwide in the genus Carex, high rates of self-pollination, high rates of chromosome rearrangement within species. So each population potentially diverging from others begs the question, do the species even cohere? Like, do we really have 2,000 species, or do we have 2 bajillion? It's hard to know for sure. So we're going to hit each of these in turn. We'll start with sedges, where the question of species coherence in the face of high chromosome diversity kind of smacks you in the face. We know sedges, right? These organisms with the closed sheaths and the 3-ranked leaves. When we're talking about sedges in this crowd, this past few days, we're talking specifically about those sedges that have perigynia. That's the genus Carex, about 2,000 species worldwide. At an estimated 2,000 species, Carex is like a nested doll of success stories. It is itself a success story globally, and within it, there are all these different success stories. When you look at this tree, this phylogenetic tree, here, a phylogenetic tree like any other, only bent around into a circle just to get it all on one page nicely. As Tony represented, if you try and do it as a line, you can't fit the whole thing on, even though you can read the names better. The colors here represent speciation rates. So globally, we see a dozen or 15 upticks in diversification in different areas of the globe. And there's no single reason for this. Every clade is special in its own way, every lineage. Like in Phacocistus, where hybridization has clearly been an important force, perhaps contributing to the diversity that we observe today. In Cobresia, appears to be some of the diversity is due to priority effects as the Himalayan Carex flora was evolving. And in the section formerly known as Ovales, chromosome evolution is clearly driving lineage diversification. Now, with all this diversity across the genus, one of the things that has struck people since the early 20th century is their high karyotypic diversity, the range of chromosome numbers that you find across the genus, a nearly continuous range from n equals 6 pairs of chromosomes to n equals 68 pairs of chromosomes. That's phenomenal. That's a whole lot of chromosomes, friends. Chromosomal evolution in the genus appears to be due primarily, almost exclusively, by fission and fusion, if you ignore the Cytorosticti, which are sister to the remainder of the genus. Why is this? Well, chromosomes in the Cyperaceae, in the Juncaceae, in butterflies, in flatworms, in a handful of clades across the tree of life are what's called holocentric. Our chromosomes have a centromere. So our chromosomes, you've seen the way the chromosome arms come down, they connect in the middle and they trail out like that. That's a typical centric chromosome. Our chromosomes are ours and the other Sedges chromosomes. The Sedges have what's called holocentric chromosomes. So if there's a breakage during meiosis, if these chromosome arms interlink like this during meiosis and one little fragment is broken off, there's all sorts of positions along the chromosome for the spindle fibers to attach. And so in centric chromosome pairs, that little fragment there that doesn't have a centromere attached to it is lost. And if there's a gene up there that codes for something important like being able to breathe, there will be no offspring as a consequence of that pairing. But when you have holocentric chromosomes and all of those fragments can migrate, what this means is that chromosome breakages are less risky. As a consequence of this, you get lots of chromosome evolution by both fission and fusion in sedges and other organisms with holocentric chromosomes. So this sets the stage for rapid chromosome evolution in Carex. And to be honest, we don't actually know why in the other holocentric groups, like rushes and like the other sedge genera, we don't see rampant agmatoploidy and symploidy, this rapid chromosome evolution. That's a mystery, but we know that it occurs in sedges and in some of the butterflies. Agrediatus butterflies are notorious for doing this as well. Okay, so this rapid chromosomal evolution may promote genetic differentiation between populations and may contribute to speciation. But that goes back to the question that we posed at the beginning. If these chromosome races, as has been argued in some work, like this excellent work by Richard Whitkiss in the 1980s and early 90s, that every chromosome race within a species could potentially be its own species, then how many Carex species are there really? Freaks you out, doesn't it? So let's look at Carex scoparia, which I look at because Paul Rothrock, you may have heard. Did you hear about this time Paul Rothrock called me on the phone? Anyway, Paul Rothrock calls me in 2000s and says, let's start looking at a few species. He had a Carex tenera problem in mind at the time. We moved on to Scoparia shortly thereafter. Paul had done this driving trip, which you may have heard about as well, going around Eastern North America, picking up Carex scoparia in all its morphological and chromosomal beauty, and making chromosome counts from around the country, and had noticed already that the morphology and the chromosomes didn't line up. So we said, well, here's a natural question. Do the genes line up with the chromosomes? Is there something happening where we're getting cryptic speciation within Carex scoparia? Again, which would be upsetting. So we had questions that were driving us because it would mean maybe that we had all these species that we couldn't recognize at all. That would be a drag. So we have questions that are driving this study. First of all, is Carex scoparia genetically cohesive? Or conversely, same question, other side, are there deep species-like genetic breaks within the species corresponding to chromosome races? Do geographically distant populations differ from each other genetically? Do changes in chromosome number drive genetic differentiation? And should we think of Carex scoparia and other karyotypically diverse species as patchworks of cryptic population-sized species? So our methods, I won't dwell on methods in this talk, but I did want to talk about AFLPs just because it's Dr. Rothrock who got me into this in the first place. Paul had collected these plants from 35 populations of Carex scoparia, variety scoparia, and Carex scoparia variety tessellata, what is now Carex wapanakakensis, a total of 48 individuals across the range. And we gathered these data sets. We gathered morphology, which we won't show the morphological data here, geographic coordinates, chromosome counts by Dr. Rothrock, and anonymous genotypic data using both AFLPs and microsatellites. And those were generated in our lab by Jamie Weber and Marcial Escudero. You'll see photos of them later. So let me just talk briefly about the AFLP methods, because just to give you a flavor of what the data are like underneath this. This is not the method we use anymore. This is a 1990s method that worked real well, and it was a big part of what we were doing for a decade, from graduate school till about 2010. What you do is you take your DNA and you chop it up with restriction enzymes. They're like molecular scissors, right? They cut the genome at specific regions that are replicable. The sequence that they attach to is specific. So when you cut a given individual's DNA twice, you should get the same array of fragments, barring sampling errors. You then amplify a subset of those using PCR. Polymerase chain reaction takes one copy of DNA. It puts complementary DNA sequences at both ends, and it builds DNA strands, and then it duplicates those and it duplicates those. So after you've done that 30 times or so, you've got about 1 million strands where you had one. And then you take all those strands and you look at their lengths by visualizing them on a gel, where short fragments run more rapidly towards the end than long fragments. And a gel looks like this. This is a digitally recreated gel where the blue bands and the yellow bands represent DNA fragments. From the longest down here to the shortest up here of individuals, where each column is one individual. And the red bands are bands of known sizes. So we could say exactly what size the DNA fragments were. We're about 2/3 of the way through the methods, don't worry. And each column then is scored. Those presences and absences, each in a size, in number of base pairs, is scored. And those then give us all these bands that each correspond to an individual. What we use those for then is as a proxy for whether a given allele, a given gene copy, is present within an individual. And I know what you're saying, Andrew, that's a crummy marker for diploid organisms. It turns out that in sedges, it's not so bad, because sedges are strongly homozygous. They tend to not have a lot of diversity at each gene copy because they're so highly selfing. That's not the reason I chose this marker. I chose it because I knew about it. But in retrospect, it turned out to be okay. So what kind of data do you get out of this process? Each ID you get a matrix of ones and zeros, kind of exciting. You take a lot of biological beauty and you boil it down to ones and zeros. Here, each ID indicates a single individual that Paul sampled in the field. Actually, these are Quercus names. This is from a Quercus talk, but that was the Carex scan that you saw earlier. Each column header represents the size of a genetic marker, one AFLP band that we represent as being either the presence or absence, 1 or 0, of a given allele. And then you can use these data to estimate genetic distances between individuals. And here we see the distance between species is higher than the distance within species. Okay, boom. That's all for that. So now what did we find using these data? First of all, we took these data and we created something that looks like a phylogenetic tree. Just think of this as a representation of genetic similarity between individuals, not as an evolutionary history so much as a proxy for genetic distance. And what we see on the right here we've graphed the chromosome numbers that Paul recovered from his observations, ranging from 2n equals 58 to 2n equals 70. Along this axis. And then the similarity between individuals is represented by their closeness on this tree. And what you see is that there are no deep breaks within Carex scoparia, according with chromosome numbers. You don't see a cluster here that represents all the 2n equals 64 individuals, right? So there's not like, we don't have chromosome races that are all acting like little species. So first of all, we're safe on that account. Second of all, there is a deep break between the two things that were formerly recognized as varieties. So Carex scoparia and Carex wapanakacensis. Carex wapanakacensis was named by Haynes and Lovett in 2012, and then studied by Marcial Escudero in 2019 in our lab using microsats. As a fun side note, the estimates on the divergence time between these is amazingly young. Certainly Pleistocene, maybe Holocene. So this is a really young divergence, one of the youngest ones documented in the plant world. And you can see, satisfyingly, genetically, the individuals from Maine of Carex scoparia are genetically dissimilar from the individuals of Carex wapanakacensis, which is also from Maine. So when those two species are co-occurring, genetically they remain distinct from one another, despite the fact that they're very, very recently diverged, maybe on the orders of a few thousand of years ago. Okay, so that was the first major finding. The second finding is that within Carex, so we've got those species separated, but we've still got all this chromosomal diversity within Carex scoparia. Within scoparia, genetic distance within individuals is correlated with geographic distance. Not that surprising, okay? We expect to see that. That's isolation by distance. When populations are far from each other, they've had a long time to diverge from each other, and they're also exchanging genes less. But the interesting third find was that when we related genetic distance to chromosome number difference, just the number of chromosomes differentiating two individuals, we also saw an effect, even when we corrected for geographic distance. And this showed up despite the fact that chromosome number difference and geographic distance are not correlated with one another. Okay, so two populations that are distant from each other and two populations that differ by a lot of chromosomes, in chromosome number, are likely to be different from each other genetically. And remarkably, this relationship between chromosome number and genetic distance, chromosome number difference and genetic distance, holds up on geographic scales from 200 to 2,200 kilometers. So what mechanism is at play? Well, here's a description of from the mammal literature where they've looked a lot at chromosome fissions and fusions as explaining speciation. Imagine that you have an ancestral population with just 3 chromosome pairs. Now, it produces a daughter population with just 2 chromosome pairs by fusion of 1 chromosome pair. That daughter population can still cross with the parent population, no problem, because at meiosis, these two chromosomes, will pair with that one, and you get a viable offspring. The same thing happens in a second offspring population, where you get fusion of two different chromosome pairs. But what happens between these two populations? They cannot cross, or not cross with very high reliability, because you get this sort of train wreck of chromosomes in meiosis. So if we see one chromosome rearrangement within sedges, we expect not to have a lot of problems. And so Martial is the older one here. He was a postdoc in our lab for many years. First came as a graduate student from Seville, came back as a postdoc, and has stayed on as a friend and colleague. He did a really excruciating germination study where he emasculated all these Carex scoparias, just sat there with forceps, like taking out the male flowers and then crossing between them, verifying that the offspring were actually the crosses he thought they were, and then looking at the effects of those crosses between different fixed chromosome numbers to see what effect that had on germination. And it turns out that the number of chromosome rearrangements has an effect on the viability, on the germination rate of the seeds from the F1s. He also ended up using this population to do a wonderful genetic map that I'll show you some other time if you're interested. So what this suggests is this model. We measured three things in this study, right? Distance between populations in kilometers, distance between populations in chromosome number, so the chromosome number difference between them, and their genetic distance. And we found out that both of these are predictors of genetic distance. We don't think this is all that's going on. We think that time is at play, right? We weren't directly measuring the amount of time between populations diverging, but time affects both the distance between populations, because of the probability of dispersal, and the genetic distance between populations because of neutral drift. And it also affects rearrangements of chromosomes. And rearrangements of chromosomes themselves affect the difference in chromosome number between two individuals and the genetic distance. And this thing that we measured here, I think, is actually a little spurious thing. We were actually measuring karyotype rearrangements indirectly, but the best we could do given the technology we had. And so when we look at, this is an imagined scenario here, but when we look at Carex across the population, imagine that each of these bubbles, each of these pumpkins up here is a Carex population with a chromosome number. And imagine that chromosome number is fixed within the population. I'll show you in a moment that that's problematic. The rate of gene flow between those populations is a function both of how far away they are from each other and their chromosome number. So you can get genes moving throughout this population, especially if there's selection, especially if there are genes that code for something that's useful to Carex scoparia across its range, moving from this population of 2n equals 60 to this population of 2n equals 70 by moving through intermediate populations. And in this way, species cohere genetically. Interestingly enough, we came back and looked at this a little bit more deeply, looking at the variation within populations. And Martial found that when we reconstruct migrations between populations, we find gene flow events between populations more than 100 kilometers apart. Not at all uncommon. Now, how much this has to do with humans moving across the landscape, we don't know, because we're not quantifying that. But this does belie the notion, it defies the notion that you should use fixed chromosomal differences between populations as justification and a blocking justification for moving seeds around on the population. And this has been argued by Frankham et al. If you were to use the criterion, don't put populations together if they have different chromosome numbers. This may matter in organisms where there's evolution by polyploidy changes. But within Carex, you would be immobilized. You wouldn't be able to do any work. And our data suggests that you don't have to worry about it that much. So you can sleep well tonight. Okay, our conclusions from this study. First of all, Carex scoparia is genetically coherent. There's no deep genetic breaks in the species that correlate with karyotypic diversity. So when people ask you, they say, look, how can you believe there's only 2,000, as you probably get asked, how can you believe there's only 2,000 species of carex when there's so much chromosomal diversity? You can say there's still movement of genes among chromosome races within sedges. There are no chromosome races. There's just chromosomal variation within the species. Karyotype rearrangements occur rapidly and form a patchwork distribution of chromosome numbers on the landscape. Gene flow is a function of both geographic distance and degree of karyotype rearrangements. And finally, sedges are good species in spite of all this nonsense. And chromosome evolution may, in fact, be part of the reason we are blessed with so many carex species. And because you guys are all in the room, I believe you think we are blessed by chromosome diversity in sedges. Okay, so now that's just the chromosome story in sedges. It's complemented by the gene flow story in oaks, this worst case scenario for the biological species concept from Coyne and Orr, who weren't botanists. So when we look at oaks are these great big outcrossing organisms. Here I'm standing in front of, I tossed this photo in based on our conversation last night about the fact that there exist at least 2 oaks in the world, the French ones and the American ones. This is one of the French white oaks. This is Quercus petrea, the one that is favored for wine barrels. The secondary compounds of this thing, you walk into a forest of Quercus petrea that's been freshly cut, and the smell is magnificent, especially if it's been raining. This individual was planted under Jean-Baptiste Colbert in around 1680 under Louis XIV. He was making plans for the French Navy. And by the time these things got big enough to build ships, they weren't building ships out of wood anymore, but they said, we need more wine barrels. So that's what these things go for. And so they're toasted like this and built into these wonderful, great big things like we were sitting next to last night. This is from a cooperage outside of Bordeaux. Anywho, it's long been recognized since the 1700s, at least, that oaks appeared to hybridize, right? And people get out onto the landscape, and they notice that when you've got, like Mike Nee noticed at his farm, when you've got 2 species that are closely related, it's not that uncommon to find intermediates. You don't find F1s very often, but you can find them. And there's enough character intermediacy in oaks that people have long been suspicious that there was hybridization going on. This was well enough known by the 70s that Lee Van Valen wrote a really important paper about the nature of ecological species, revolving around some work that Bill Berger had done in white oaks, in which he wrote, it may well be that Quercus macrocarpa in Quebec exchanges many more genes with local bicolor. So bur oak exchanges more genes with local swamp white oak than it does with bur oak in Texas, right? Which would be scandalizing if we were to, in fact, we could say exactly the same thing about modern humans and Neanderthals if we were talking, 40,000 years ago. We could have been. So we now know the gene flow, based on all the research that's been done over the past 30 years, it's a key part of oak evolution at local scales, at continental scales, and at global scales. And in fact, it's been going on as long as there have been oaks around. This kind of philandering between species is part of what makes oaks what they are. So promiscuity is a central part of the oak identity. So when this is going on, people always have worries about, like, what do we mean by oak species, and are oaks genetically coherent? Do we need to think about oaks as not being genetically coherent in the same way that we think of sedges? So in this part of the talk, I'll focus on 2 case studies, one in the red oak group, one in the white oak group, that address 3 overarching questions. First of all, does species identity, the name you put on a species, given what its morphology tells you, explain genetic diversity within oaks? Because if not, all bets are off, or at least a subset of bets. Second, is genotype better explained by geography or climatic and ecological site similarity? It could be that you're able to explain some genetic diversity by species identity, but there's an awful lot still explained by geography. And does genetic similarity between populations within species swamp gene flow between species? Are there regions of the genome that define oak species? Okay, so the Western Great Lakes region offers us a great set of species for studying these questions. I'm going to show you first, this first study has been work that we've been doing since I first started at the Arboretum. I was a sedge guy, came to the Arboretum. They said, you've got to work on some woody plants if you want to last here. I said, I'll work on oaks. And so I was suggested that I should work on Quercus ellipsoidalis and allies. It's long been entangled with scarlet oak. Both of them can be confused with black oak, which encompasses most of the combined range. I'm mostly going to focus on the interaction between Hills oak and black oak, which is relevant to this question. Leave scarlet oak for another time, because that's more complicated than I believed it was several years ago. It had been known morphologically that Hills oak and black oak cross. This is some of Dick Jensen's foundational and really nicely done work from the 1970s. Based on morphology. Here is black oak with its nice fringy acorns, its densely canescent end buds. And here's Hills oak with its imbricated buds, bud scales, and its more deeply divided leaves. I don't have the end buds here. Silky pubescent typically. Our sampling for this study was about 851 individuals from 58 different sites. And we used AFLPs for this study. I won't dwell on what AFLPs are because you know already. Here we analyzed them differently. What you're looking at here, this figure shows each individual is a column on this figure. The color of the column estimates the genotypic composition of that individual. So an individual that has a single color, we suggest that that individual has sort of a homogeneous genome with respect to some ancestral populations that we hypothesize. And an individual that has high variation shows some admixture or mixing between populations. What the mechanism underlying that isn't defined by the analysis, but it's usually assumed to be gene flow. Okay, so at the outset, one thing we notice is that there's a little gene flow between species. There are some individuals that look like F1s, like these individuals of Quercus coccinea here, right? That look like they're mixed between coccinea and ellipsoidalis, a problem worth coming back to. There are some out-and-out misclassifications, things that we identify positively. We threw all our questionables over here. These are things that morphologically we couldn't be confident of because the material was inadequate. But stuff where we had good acorns, good end buds, good leaves, sometimes you misidentify things. Of the three species then, Quercus ellipsoidalis, the Hills oak, exhibits the greatest genetic variation. There's the most colors within it, where each individual is a species. Does this mean the species is not genetically coherent? When we throw those genotypes onto the map, this is a map that was done by Marlene Hahn. You'll get to see her in a little bit, not in person. Each of these pies, the colors of the pies, indicate what the genotypic composition of individuals at a site was. And what it shows is that the genotypic composition of these individuals in the upper portion of the lower peninsula of Michigan are different than the ones from the Chicago region. So are they a different thing? No. It turns out that when you cluster them, when you throw them together genetically, they do not cluster by geography. You don't get a black oak from Berrien County or from Cook County with a Hills oak from Cook County, and separating out by geography like that. What you find is that all the Hills oak clustered together, all the black oak clustered together, and insofar as we sample them, all the red oaks and all the Quercus coccinea, all the scarlet oaks clustered together as species. So this idea that Lee Van Balen had thrown out there, that genes swap more readily between individuals at a site than they do between individuals of a given species across numerous sites, even if that's true, it doesn't subvert our genotypic picture of what a species is, which again is kind of satisfying. But let's let that be true for a moment and say that even if these are good species in that genotypic sense, which accords closely with Darwin's notion of what a species was morphologically, two things that living together, two entities, populations that living together, retain their distinctiveness for generations. Could geography and ecology still explain an important component of this genetic similarity? So here we modeled genetic diversity. Again, here we've got the distance between individuals or populations in AFLP distance. as a function of whether or not they differ in identity, what we identified them as, whether they differ, whether they're geographically widespread from one another, and whether they're ecologically widespread. Here we used soil and climatic layers to estimate ecological dissimilarity. And there's the data we used. And it turns out that when we look at all of the species together, black oak, hills oak, and scarlet oak, difference in identity, difference in the name you put on a species explains about 30% of the variation. And ecological distance explains about 6%. Distance explains nothing. And when you just look at black and hills oak, just those two, the name you put on the species based on its morphology, primarily. These analyses were actually conducted using both genetics and morphology. The analysis is the same with morphology, and that's how it'll be done for publication. Identity explains 34% of the variation, whereas distance only explains 2%. Ecology only explains about 6%. Okay, so genetically, these things are good species, despite the gene flow that goes on between them. But as we know, we can't use genes to identify species in the field. We have to know how these correlate with morphology if we're going to have sensible application of genetic data to field identification questions. So how does the morphological variation relate to genetic variation? So here, Marlena, who was just starting in my lab at the time that we did this project, led a morphological study of, I think, about 150 of these individuals, looking at both fringiness of the acorn cap. So she created this jig to relate sort of how fringed an acorn cap was from the fringiest, representing what we expect in Quercus volutna, the black oak, to the least fringy, what we expect in hills oak. We looked at the trichomes on the inner surface of the acorn cap, which are very good for distinguishing black oak from hills oak, by running transects down these acorn caps and measuring visually what the trichome density was along these transects, and the length and pubescence on the end buds. Using a little jig that we could use as, again, they were like little quadrats running down the end buds, saying in how many of them can I see versus not see the scales through the hairs. When you look at each of these characters alone, where the black bars represent Quercus volutna, black oak individuals, and the red bars represent hills oak individuals, you see that none of the characters alone does the trick. But when you put them into multivariate space, you take all four of these characters and you crush them down into 2 dimensions using ordination, and you overlay on them the genotypes. The genotypes here are represented by the shapes and the colors, where the black squares are Quercus volutna genotypes, the red circles are Quercus ellipsoidalis genotypes, and the size of the character represents how confident we are. The smallest characters, these little gray diamonds, those are the ones that are hybrid genotypes. We can't tell what we're looking at. And what it turns out that we can figure out from the morphology is a hell of a lot. Usually, we know about 95% of the time when we look at an individual and we key it out as Quercus ellipsoidalis, we're correct. Or Quercus volutna, we're correct. But there are these handful of individuals here that appear morphologically to be Quercus volutna and are genetically intermediate. I'm sorry. And this handful where we key them out and they appear to be Quercus ellipsoidalis and they're genetically intermediate. And this small handful of individuals here in the middle that are genetically intermediate, morphologically intermediate, but genetically one or the other. There is a little bit of discord, and that's a hallmark of intergressive hybridization. It's exactly what Edgar Anderson predicted in 1949. Is that the interesting cases in hybridization are not the F1s, not the bizarro first generation hybrids. They're these cases where there's gene flow ongoing, slippage of genes from one species to another with lots and lots of backcrossing, which is certainly what we see in Quercus, and maintenance of those genes that matter for the ecology of the individual by natural selection. And that's what we got going on in oaks. Despite that fact, we still have good species with a little bit of uncertainty. And that kind of uncertainty, there's nothing we can do about. We have to live with it. But fortunately, we can get data about it. So what conclusions can we raise from this? First of all, individuals form genetically distinct clusters by species, not by region or by site. There's more evidence of admixture between black oak and Hills oak than between black oak and scarlet oak. And there's limited evidence of admixture between scarlet oak and Hills oak. And despite evidence of this admixture, morphological species identification and molecular population assignments correlate well when adequate material is available. And I want to close this sermon with just one more, this talk, sorry. I did grow up in the church. And so so I still think of it. With one more case study, more recent, where we went back and looked at a really cool study by James Harden in 1975. James Harden published this wonderful article in the Journal of the Arnold Arboretum, where he displayed he displayed 16 of the Eastern North American white oaks, and each line between species represents an unnatural act, a pairing between species that was documented either experimentally or by inspection of morphological variants in the field or in the herbarium. And he said, look, all the white oaks are doing it with somebody else besides their own species, with the exception of Chapmanii and Oglethorpensis, and even they're outed at this point. They're mixed up with somebody. So in our study, we investigated 7 species from Harden's 1975 oak syngamion paper, where the syngamion is syn for together, gamion for gene flow. It's this group of species that retain their species distinctions despite the fact that there's gene flow between them. It was a term it was termed by Lotze in 1916 and again in 1925. Lotze also, however, didn't believe in species. Don't have to, you're not obliged. Quercus alba, Quercus macrocarpa, Quercus bicolor, Montana, Michoii, Muhlenbergii, and Prinoides here mixed because genetically we can't tell them apart. I believe they're species in the ecological sense. We don't know yet what they are. And Quercus stellata, post oak. So what did we do here? Here we're like, okay, we're moving on beyond AFLPs. We had gathered, by the time we did this study in 2019, a huge phylogenomic data set of lots of oaks from around the world, and we were able to use these to create a DNA barcode. But first we needed samples. So Mira Garner, who had started in our lab as an undergraduate in 2015, came back and worked on our Carex project, came back and worked on Quercus, and is now hired for another 3 years, 4 years? 3 years right now on a big Quercus macrocarpa project, cruise all around the country, collecting Quercus macrocarpa and all the co-occurring white oaks that she could find with those Quercus macrocarpa. Okay, so wonderful road trip, set of road trips. We then developed a DNA barcode in the very narrow, in a very broad sense of the term, developed by a postdoc in my lab, Elizabeth Fitzek, on a grant to go work at INRA Biogico in Bordeaux, where we have colleagues where I'd done a sabbatical, and created a barcode of 80 single nucleotide polymorphisms from across the genotype. So it's something you can do cheap and dirty. It's like an $11 genotype for identifying a bunch of Eastern North American oaks. We selected from this phylogenomic data set just SNPs that were variable between species with low variation within species. So you wouldn't want to use these markers to study introgression per se, because they're biased towards not detecting introgression, but they're very good for detecting F1 hybrids and things like that. So we analyzed these data first using a clustering approach, UPGMA, that places individuals in groupings just based on their genetic similarity. And when you look at these, the long branches leading to each species are indicative of high genetic differences between species, low variation within, which is exactly what you expect with the particular markers we developed. So this underestimates the genetic variation within species, but because these markers were developed on one set of individuals and then genotyped on a very broad set of individuals, it validates that we're finding genomic markers that actually indicate genetic coherence of these species across their range, despite all the interbreeding that Harden had documented. Moreover, when we use the clustering approach I showed you earlier with all these bars, here I'm going to show you just a set of diagrams. What I want you to do is focus on the one at the right near the bottom, the k equals 8. What we're doing here is each column is an individual. The colors of that column indicate what its genotypic composition is. relative to a postulated set of ancestral populations, whether it's 2 populations or 9 populations or anything in between. When we get up to 8 populations, we can distinguish species from each other exceptionally well, right? Except for Carex prinoides and Muhlenbergii, which again, we don't know what's going on there. But we also document a level of gene flow between individuals, despite the fact that the markers that we selected were selected against, they were choosing markers that should be less susceptible to introgression. So here we've recreated Hardin's illustration, but just for the 7 species that we worked on. The thickness of each line indicates a coarse estimate of what gene flow might be between those individuals. So that despite the fact that we can recognize these species across the range, they are a true syngamion with gene flow going on at some level all the time. So in Punchline, when you take these data, along with lots of other data, there's been so much exciting work on this in the last 2 decades. Even if it's the case, as Lee Van Valen suggested, that local white oaks and local bur oaks are swapping more genes with each other than the white oak in Texas, than the bur oak in Texas is with the bur oak in Illinois. There is enough genetic similarity between populations, due perhaps to migration of genes that are under strong selection, due perhaps to inherited ancestry and subsequent stabilizing selection. We don't know what the mechanism is yet. Those markers, those regions of the genome, are what make an oak species an oak species. So when we talk about oak species, despite the fact that there's just gene flow that should screw up our notion of species, there are regions of the genome that define oaks. There is, however, a wrinkle. So this is we've mapped markers back to the genome, back to the 12 chromosome pairs of oaks. And without belaboring how you read this and what this says, the punchline here is that when we map these markers from different lineages, where we've studied introgression versus divergence, introgression, gene flow between species, divergence, separation of species over time, it turns out that the genes that define species in one lineage are not the same as the genes that define a lineage in another species. So when we talk about oak species, we're not talking about like one set of genes that make an oak. What it is this entire genomic background under strong ecological selection at the suite of species we're looking at and in the region where we're looking. So the history of oaks is not unitary. The oak genome, like the human genome, is a mosaic of oak histories, whether you're looking at Quercus gambellii or you're looking at modern humans. All these stories are embedded within our genomes, racing around. And when we look at sedges, it's exactly the same. So what can the proper, the sedges, and the promiscuous, the oaks, tell us in combination? First, species are dynamic entities. They cohere genetically, ecologically, and morphologically across broad geographic ranges. They are things that you can believe in, which is a very old-fashioned thing for me to say. But when I say species, I believe strongly that if you don't believe in a thing, you shouldn't talk about it all that much. And so if I didn't believe that species were a thing, I shouldn't say species. But I suspect that when we say species, we're talking about something meaningful on the landscape, to which you might say, not a big deal, Andrew, we knew that. But it is something we have to ask about when there's all this gene flow that subverts our idea of what the hell a species is. Second of all, species boundaries are often, almost always, semi-permeable, with species cohering and remaining distinct from one another despite ongoing introgressive hybridization. And when barriers to gene flow evolve rapidly, like they do in sedges, stepwise relationships, iterative, accruing changes between populations may still allow species coherence. Right? So we can believe in species, but they're going to be complicated. And finally, when we say species, we're not talking about the outcome of a single process, the genes at a single locus. Again, I would say when we say species, we usually mean what Darwin meant when in The Origin of Species, when he says, what the hell are species? He says, if you want to diagnose species, look for populations that have been living together for hundreds of generations if you can. Right? And are they still distinct despite some ongoing gene flow? If they are, then that's probably what a species is. Don't tie your species notion up with any particular process. You can then go back and say, why are they species? And that's going to vary across the tree of life, whether you're looking at bacteria or looking at sedges or you're looking at oaks, even ants. I couldn't have done this work without a lot of colleagues, collaborators in various aspects of the work presented here. When I tried to make a list, it took up the slide. Support from the Arboretum, USDA, and U.S. National Arboretum, National Science Foundation, the Fulbright Commission, my lab, who make my life wonderful. I mean, my wonderful life. Just they make life livable. And they're friends, they're colleagues. We work together and we learn from each other. The many, many friends and colleagues of the Global Carex Group. This is a photo from our kickoff meeting in 2011, where we got together individuals from around the world who worked on Carex with individuals from the upper Midwest who were focused on Carex to try and effect a kind of a synthesis between a synthesis of the global classification and one that would be useful to practitioners in the field so that we could move beyond having an academic practitioner divide and have a Carex working group. The folks I work with on oaks, who again, it just seemed to be increasing in number. We were a small group working on the American oaks, and then we grew in number to work on the oaks of the world, and we're growing again to work on the oaks of Mexico. My mentors. We're all lucky to have people in our lives who've taught us things and put up with our bullshit. And Tony and Paul are both. They have taught me so much and continue to teach me. And I'm very grateful. I wouldn't be here if it weren't for many, many people, many chance conversations, many people giving me opportunities when they should not have done so. Thank you, Tony and Paul. I don't know if it was a good decision, but I'm grateful. And finally, thank you all. You guys could have been anywhere. I've just being here this last few days, I keep thinking of David Foster Wallace writing about tennis, and he talks about watching these brilliant tennis players, and he's there making notes and remembering like his own tennis games and feeling like, oh my God, these guys really know how to play tennis. That's how I feel in the field and in conversation with people in this room is like, I feel like, I guess I kind of know how to play tennis, but these guys really know how to play. So thank you. This has been a ton of fun. Let's take a few minutes for questions because I don't want to hold up lunch, but I'll take a couple of minutes for questions. Thank you.
So Andrew, I've been sent here by the Me Too movement. When you talk about all the miscegenation between species and oaks, have you identified whether it's the males or the females that are primarily responsible for that?
Well, we know a lot about sex, and I don't think we can blame the males or the females. They're both involved, as is often the case with these things. So when an oak loves another oak very much, the male produces the pollen, and the pollen is out there in the world, right? And the oak stigma, the female, the pistillate flower is receptive, and the pollen grains land on there. And one of the great things that happens in the Phagales, this order that includes oaks and includes the walnuts, includes the birches, includes the Nothofagaceae, the southern beeches, and a few other families, they all have delayed fertilization. So the pollen grains land there and they germinate just a little bit, and then they hang out for a couple of weeks to a couple of months before they go down and finish off their fertilization of the ovule. During that time, if you get multiple species landing on that stigma, and if one of the species is the species that accords with the mother plant's own species, it tends to be the case that all of the pollen tubes are aborted except for the one or ones that accord with her own species. So there is a selective process that goes on. So like the worst way to get a hybrid is to stick a bunch of oaks together and grow them in an intermixed patch and grow like evenly distributed Quercus alba, Quercus macrocarp, Quercus bicolor, because there's going to be lots of pollen landing on every stigma, and there's going to be lots of selection going on, and the moms are going to tend to do fairly well. But if you put one Quercus macrocarpa in an ocean of Quercus alba, what's going to happen? She's going to be pollinated by Quercus alba. So who's to blame? I think there's blame to go around. but that's the process by which it happens. Credit. There's credit. Let's give credit where credit is due. Does that answer your question, Eric?
It was more, once you get past the F1 hybrid, when you start getting into the progression going back into species, is there a clear male bias or female bias as far as how that progression proceeds?
That's a great question. If it's governed by anything other than abundance of individuals on the landscape, it's not clear to me. We haven't done that research ourselves, but looking at the literature, what I see is lots and lots that has to do with abundance on the landscape and tendency of pollen and seeds to move. There's a wonderful article from 2003 by Antoine Kremer et al. In New Phytologist about hybridization as a mechanism for migration in oaks, where they show pretty convincingly, I didn't believe it for many years, but I do now, that Quercus, two of the French oaks, that Quercus robur and Petreas, they were marching north following the recession of the glaciers, that they moved by piggybacking, where one tossed its seeds out, and I can't remember who has the tastier seed, more dispersible seed. Those would get established, and then it would be swamped by pollen of the other species. And so by hybridization, turn over to the other species. So again, it seems to be that it has more to do with life history and abundance on the landscape than anything else. There could be other mechanisms at play as well, but I don't know of them. Thank you all very much.